My name is Yuan and my scientist is Janet Iwasa. Janet Iwasa was born in 1978, Bloomington, Indiana. Growing up in Maryland, she would go with her father to his lab at the National Institutes of Health, where he worked as a physics scientist. This would act as the main source of inspiration for Iwasa to become a scientist herself. While her father worked in his lab, she would go on his Apple Macintosh and draw for hours. In high school, Iwasa became properly involved in the scientific field when she took part in an internship at the Institute of Genomic Research. After graduating from Williams College, Iwasa would go on to earn her PhD from the University of California. During her years as a graduate student, she took animation classes and training programs to acquire the skills needed to animate molecular shapes and movements. What I admire most about Janet Iwasa is her dedication to her craft. Her achievements in receiving a PhD and becoming a professional animator helped provide visuals that raised questions, discovered new information, and encouraged communication of the science behind the research. As someone that has animated before, I can recognize and relate to the effort that goes into presenting an idea through animation. Iwasa is mainly involved in biochemistry, as well as focusing on cell biology and molecular animation. At the center of her work is how animation can be used to further our understanding of molecules. Iwasa's most recent experiment, titled Phosphorylation of the ARP23 complex is necessary to nucleate actin filaments, attempts to find the process required to bind and nucleate actin filaments. To rehydrate the ARP23 complex, chloroform and methanol extractions were precipitated onto it and then exposed to a rehydration buffer. The proteins were separated in the first and second dimension using an Eton IPG4-3IEF system and new PAGE Bistris precast gels. 12% SDS PAGE was used to separate the ARP23 complex and it is then dephosphorylated with AP. ARP23's binding constants were determined using GST-SCAR VCA and GST-NWASP. Proteins were digested after reduction and alkylation. The images for the figures were captured using a Roper SPOT charge-coupled camera on an Axophone microscope. So far, Iwasa's animations have helped scientists bring various molecular actions to life: cell ingestion, protein transportation, motor proteins and cell division, and endoses are just a few complex models she has worked on. With her animations, we are able to understand how HIV enters and exits human immune cells and an HIV cell's complete life cycle. Iwasa is currently still developing visual models that represent various molecular processes. And here are the references.